Hello lovelies, in this video we're going to be looking at plant responses for your A-level biology.
Hi everyone. So we looked at animal responses, which we described into two groups known as taxes and kineses. And now I'm going to move on to looking at the similar responses that happen in plants, except in this case, we're talking about tropisms. Now, tropisms are very similar or mostly similar to taxes, and they're just called something slightly different. And we'll explain why mostly. It's because, as we said in the previous video, plants can't move away or towards a stimulus. They can't get up and physically move themselves. They're not mobile organisms, but they can change the way they grow in response to changes in their environment. So if it's a movement towards or away from a stimulus that involves the whole organism moving, then that is then a taxis. If it's something where a plant is obviously just changing the direction of growth towards or away from a stimulus, then it's a tropism. And in the same way as the other responses, the taxes and kineses, these can help plants increase their chances of survival. So for things that plants need, again, before it was thinking about predators, food, mates, going towards resources that they need, similar for plants. So obviously they need more light, because if they have more light, they can do more photosynthesis. If they have more water and minerals, obviously they need minerals, various minerals for growth. And more stability. So I'm thinking about roots in the ground, so having a greater kind of connection to the soil and therefore holding their own if it's windy, if it's very exposed, if they're likely to be pulled up very easily or knocked over very easily, and therefore if they were on the ground they would get less light, so being able to be supported is very important. So we're going to look at how different tropisms can help them achieve this. They are directional. So they are still directional, which is why they are a tropism. And I'll talk briefly about the other kind of plant response that's non-directional, although you don't need to learn that for this specification. So they either grow towards or away from the stimulus. So that's the same thing as before. So towards will be a positive tropism, whereas away will be a negative tropism. But just in case, make sure this time we're talking about growth towards or away. We're not talking about movement towards or away, because that would suggest the organism is physically moving. Okay, so in the same way with taxis, the prefix in front of the tropism describes the stimulus that causes it. And again, remember, we can have positive and negative tropisms describing the movement towards or away. So we're going to have a look at 4 examples of plant tropisms. Again, most of these it would be good to learn because you are going to be given these as examples. There aren't many tropic responses in plants, so the more you can learn, the better you'll be able to identify them and then justify them. So the first one is probably the most obvious one, phototropism. So photo, again, that prefix meaning light. So we're going to talk about shoots and roots as separate entities when we talk about these tropisms, because they often have different responses. So shoots are positively phototropic. They grow towards the light, and that makes sense because they want to maximize the light for photosynthesis. Roots are negatively phototropic, so they grow away from the light, which encourages them to grow down into the ground. That makes more sense. There are no chloroplasts in roots, and they need to grow down because that means that they'll be growing away from the light, which suggests they're growing into the soil, and that's where they're going to need to grow to get more water and nutrients, etc. So then we have geotropism, sometimes also called gravitropism. So geotropism or gravitropism is the growth towards or away from the field of gravity or the gravitational pull. So shoots are negatively geotropic or gravitropic. They grow against the gravitational field. They make sure they're growing above the ground towards the sky, so away from the center of the earth, to make sure that there's more light, because again, they need more light for photosynthesis. So if you were growing with gravity, you'd be growing into the ground, which is not what we want if we are a shoot. Roots are then the opposite. They are positively geotropic or gravitropic. That means they grow down into the ground, and the deeper they go, the more likely they are to find more water, mineral ions, and also roots being sort of deep into the ground helps to stabilize the plant as well. So it means it's less likely to fall over, get knocked over in high winds. It gives this stability. So we've got negatively geotropic shoots and positively geotropic roots. Okay, so there are some other plant responses that are non-directional. So some of these are called nastic responses. So they're responses that the plant obviously takes in from its environment and there'll be some kind of stimulus and then it's responding in a certain way, but this time it's just not in a specific direction towards or away from the stimulus. So it can be changes in turgor pressure, so changing the pressure and how full water is in cells to shrink or swell those tissues, or it could be growth responses, or it can be things like moving the leaves and stuff like that. So that's what these kind of responses kind of carry out. So they could be in response to a biotic or an abiotic stress. So a lot of these, for example, are abiotic stress conditions or biotic stress conditions. So flowers and leaves closing at night in response to temperature, that's obviously an abiotic stress. If it gets cold overnight, potential for frost, they're going to close their leaves up. Shutting of a Venus flytrap, that's in response to the stress from nutrients and needing extra nutrients, and then detecting the stimulus of a fly or something they can use to actually get more nutrients. And then you can see my mimosa plant here. This is actually a defense response to herbivory. So that's a biotic stress, and it's closing its leaves there when it gets touched. So if it gets touched, it will close its leaves up and actually pull them in closer to the stems. And that's really kind of useful and important, because what it will do is it will kind of potentially scare the animal because they're not expecting a plant to move. It makes it harder to actually get to the leaves, and then they're not as visible when they're all closed up. And also, if there is an insect or something on those leaves, they can fall off. So this is sort of an idea of a response to trying to avoid being eaten. There's some other responses that plants can do that are also chemical-based defenses, which we've looked at when we looked at plant defenses as well already. So there's tannins that the plant can produce in response to potentially being attacked by a pathogen or a herbivore. So this makes the leaves taste bad. So it's trying to put the animals off from eating the leaves because they're not going to taste nice. And it also can do things like protect the roots from infection by various insects or other pathogens. There are also alkaloids, another chemical plants can produce. This again makes a bitter taste, so it becomes a feeding deterrent. If something's trying to eat these leaves, they're going to taste horrible and put the organisms off from chewing the leaves. And lastly, pheromones, which is actually really cool because it means that it's a way of plants actually communicating to other plants. So when a plant can give out these pheromone chemicals that are very volatile and can kind of blow on the wind, other plants can detect these. So if it's under attack from a specific insect or a herbivore, it can let out these pheromones to let other plants nearby know that these animals might be coming for you next. So they can do things like increase the amount of tannins and alkaloids in their leaves and tissues so that they can be protected in advance for those herbivores coming. And it's a really nice way of plants communicating with each other. So these tannins and alkaloids are going to be found in things like leaves, shoot tips, flowers, and external layers of stems and roots in order to try and make sure they're the bits that are going to be eaten by the animals first.
Okay, so plant responses are controlled by growth factors, and we use the term growth factors here, but they behave in a similar way to hormones. So they're hormone-like chemicals. They are produced at the tips of roots and shoots in the meristem. There's different classes of them, and they can travel around the plant in diffusion, active transport, or even in the vascular tissue if they need to. And we've got different groups, including auxins, gibberellins, abscisic acid, ethene. For this spec, we're just going to talk about auxins. If your spec is different and includes the others and you need more detail on that, then there will be a separate video for the action and use of those hormones. So IAA is the example we're going to look at, which is indoacetic acid, and it's an example of an auxin. So it's within the group of auxins, and it controls both photo and geotropisms or gravitropisms in both roots and shoots. So let's look at phototropism in shoots first. So the way it works is that IAA moves to the more shaded part of the shoot. So in this case, we've got the sun moving throughout the daytime and changing direction. So the sun is initially pointing straight down. So we have none of the tip is shaded any different to any of the rest of it. And then as the sun moves across the sky during the day, then we get a shaded side and a sunny side of the shoot tip. What happens is the IAA gets moved to the shaded side, and that promotes the growth of cells on that side of the shoot, the elongation of those cells. And so that causes the shoot to bend towards the light because the same elongation is not happening on the sunny side of the shoot. So we get that bending of the shoot tip towards the direction the light is in. So this is obviously a tropism. We're positively phototropic here. We're moving the growth towards the stimulus, which is the light. Okay, so moving on to negative phototropism in roots. So this is where we're obviously going to be growing away from the light, and we said that roots are negatively phototropic, so they're going to be growing away from the light because they're going down into the ground. So in this case, IAA moves to the more shaded side of the root still, but it inhibits growth. So because it inhibits growth on that side, the cells on the unshaded side elongate, whereas the shaded side does not, and therefore we're going to be bending down and away from the direction of the light. Okay, so then if we think about gravitropism or geotropism as well, it happens in the same way. So positive geotropism works in the same way as negative phototropism in roots, because the IAA is going to move to the side that is lowest to the ground or closest to that gravitational pull. Again, it's going to inhibit elongation in those cells, whereas the elongation occurs in the side that's furthest away from the gravitational pull. And so then we get that movement down, sort of bending down and growing down towards the gravitational pull. So that's how the roots kind of go further into the ground. So mainly the remembering sort of facts here is to kind of think about where is the IAA going, and if it's always moving to the shaded side or kind of the side that's pulling down with gravity. But in shoots, it's promoting the elongation, so it will bend towards the light. In roots, it's inhibiting elongation. So then we bend away from the light, or we bend down towards gravity. So that's the kind of thing to remember. So in shoots, IAA is causing elongation or promoting elongation. In roots, it's inhibiting elongation on that side, and therefore the elongation occurs on the opposite side and we get bending away. So I've condensed this down into a table so it's easy for us to remember kind of the differences here. So if we have a low concentration of IAA in a shoot, then the cell growth is inhibited. If we have a high concentration of IAA in a shoot, then the cell growth is stimulated. So obviously when all of the IAA moves over to one side, that creates a high concentration, and then we get elongation on that side. So this produces a phototropic response in shoots, which is positive, but in terms of a geotropic response in shoots, that will be negative because they tend to grow away from the pull of gravity. In roots, it's the opposite then. So in roots where you have a low concentration of IAA, then the cell growth is actually encouraged and stimulated, whereas the high concentration of IAA, then cell growth is inhibited. So we get this negative phototropic response, so they're moving in the opposite direction from the shoots. They're going down with the pull of gravity. So it's a negative phototropic response, but a positive geotropic response. Okay, so in a way to explain some of these sort of methods that auxin and IAA can work in shoots and how they cause this phototropic response, there's been experiments over many years that demonstrated the different conclusions that we now have about how IAA works. So first of all, simple experiments showing that IAA is produced in the shoot meristem and is the reason that the shoots bend towards light. So if you remove the tip and remove the meristem, there is no bending of the shoot towards light, even though it's a directional source of light. If you cover the tip with something that stops it from being able to sense the light, so you cover it in tinfoil or black paper or something, again, there's no bending towards the light. So what we can conclude from these simple experiments is that the source of the light sensing ability is located in the tip of the shoot, and therefore it controls the growth. So if you remove the ability to sense light, you remove the phototropic response and you move the growth response. And that was the sort of start to show that the tip was really important as to where maybe that the control center for this response is going on. Then we sort of had experiments that showed and demonstrated that actually it's the movement of a chemical that comes down from the tip in order to control this growth. So diffusion is important here. So by putting a physical barrier, a sheet of mica was what they used, but something that chemicals can't diffuse through, by putting that barrier on the shaded side and exposing them to a directional source of light, there's no bending. So the IA must need to be able to diffuse down from the tip, down that shaded side in order to cause the bending towards the light. And we can show that and confirm that by if you put the mica where the IA can't diffuse down the light side, you still get bending towards the light. So when the barrier is on the light side, it doesn't matter because the IA can still diffuse down from the tip down around the outside of that, down the shaded side, which is where it's causing elongation. So we get this idea that we have to have some sort of movement of a chemical from the tip down. It can't come from up. If we put that barrier there, it wouldn't be coming from below the tip. And also, it clearly can't just move around and diffuse across from one side to the other. It's definitely coming down from that apex of the meristem. And then equally, you can show the same thing as that it has to be able to diffuse by putting a barrier in this time, an even barrier, so across the whole tip, but made of agar gel. The same thing we use in gel electrophoresis, and the same thing you use when you grow microorganisms on a plate. So bending still occurs when you can have a substance like agar that the IAA can diffuse through. So you've removed the tip, but you've replaced the tip back onto some agar gel, which you've put in the middle. So even though you've separated the cells from each other, the tip is still functioning, it's still producing IAA, and that is diffusing down through the agar into the rest of the shoot, and it's causing that bending towards the light and that uneven growth. And then we need to kind of link this idea that the IAA distribution, that movement of the IAA, is causing the phototropic response. It's linked to light in some way, or it's linked, the movement of the IAA down one side or the other is linked to them being able to sense light on one side and shade on the other side. So if you physically remove the tip and place it back onto the shoot, but at an angle, so it's only on one side, it doesn't really matter what side, but you put the shoot tip in complete darkness, so there is no light there. But because we've moved the tip only to one side, IA can only diffuse down that side, because we've established it can't just move across and it's coming from the tip, then you still get bending, even in the dark when there's no light. So that must mean that this uneven distribution here is causing the bending, it's going to that side. And it's only able to do that in this example because we've put it to one side. There is no light here to sense in order to be able to grow that way, but it's still happening. Same and exactly the same thing again, and it shows the same thing. You can do the sort of the agar gel experiment, but you don't need there's no light sensing going on here, but you can cut the tips, let the IAA diffuse out of the tips into the agar gel, and then just put a cube of that agar gel that contains the IAA onto a sheet. Again, put it to one side unevenly on the top, and then even in the dark, again, the shoot will bend. So that's just showing you that it's nothing to do with the tip. There's no light sensing necessarily in the tip happening, and it can't because it's in the dark anyway. And it's pushed to one side, so you've got the IAA only being able to diffuse down one side, so it's very uneven, and that's causing this bending towards the light. So we're getting a phototropic response even though there's no light. These experiments show that the IAA concentration must be being controlled in the light. So when it is light, when the shoot tips are exposed to light, then they are able to tell where the shaded side is and where the light side is, and the movement of IAA is then organised based on where the shaded side is. And it moves down that side, and there's a higher concentration on the shaded side, which causes that elongation and bending towards the light. Because even in the dark this will happen, but only when you put the IAA source to one side and make it an uneven distribution. If we just put that agar tip on the top, the shoot would just grow upwards, which is what shoots do in the dark anyway as a response to darkness. They're trying to grow up straight as fast as they can to see if they can get some light because they think they're still underground. The main thing as well to remember is that in these experiments, you would need to think about how you're going to make sure the plant has enough glucose in order to grow, because it can't be doing photosynthesis. So if you want them to grow in the dark, you're going to need to provide them with some glucose. You can put that in the agar, for example. And also for all of these experiments, you'd need controls. So as I said, you could put the agar block in the middle in the dark experiments to show that it would just go straight up. Same, you could cut the tip and put it in the middle to show that it's going the same up. You can use agar blocks that contain water as well, a negative control, to show that it's the IAA that's present there that's having the effect. All of these things are kind of experimental conditions we'd have to think about, and these could be questions that you could see where they give you different experiments like this and say, well, what conclusions can you draw from this, or what controls should they have used, or why might they have added glucose to the agar in the dark experiments, things like that, questions like that could come up. So it's not necessarily about needing to learn these 100% off by heart. It's about being able to apply your knowledge of IAA and how it moves to kind of explain what these experiments showed and what conclusions they showed. Or if someone's done a similar experiment, to be able to explain why they've seen those responses in the shoot tips. Okay, so let's look at how you can investigate tropisms doing some practical methods that we can do in school. First up, let's look at phototropism. This is an example of a method or a technique you can use to investigate phototropism responses in plants. You have to grow shoots, but you're going to cover different parts of the plant with foil. So shoot A is uncovered, shoot B has a tinfoil hat just covering the tip of the shoot, and shoot C has foil round the middle underneath. We need a light source that's coming in a direction, because otherwise, if you just have a light source above, there'll be no bending or moving towards or away from the light. So we need a light source coming from one direction. We need to have got many seedlings, not just 3. We need lots of repeats here. And some of the seedlings you'll keep all in the same conditions. You've got to control all the growth conditions that could affect growth. So temperature, water availability, nutrient availability. And then you'll measure the responses towards the end of the experiment by measuring growth. So you should also do some measurements, so measuring the amount of growth or the height of the seedlings over time, and this way you can get some quantitative data as well as doing some observations and getting some qualitative data about the bending of the shoots. You also need to know how to investigate geotropisms. You need to put a set number of seeds in Petri dishes on damp cotton wool. There should be the same number of seeds in each dish, and each bit of cotton wool should have received the same volume of water. You're then going to place the dishes in dark, or you're going to wrap them in tinfoil to exclude light. This means that we're only going to be looking at the effect of gravity and not light. Then you need to place the Petri dishes at different angles. This could be on different blocks, taping them upside down, taping them to something that's vertical. Then you need to leave them to germinate with all other conditions exactly the same. So there should be no light for all of them. They should be receiving, again, they should have received the same volume of water. There should be the same time, same temperature. After a set amount of time, you need to record the direction of shoot and root growth for each condition. All should show positive geotropism in their roots and negative geotropism in their shoots, regardless of the angle they were grown at.
Ouch! This is why in some videos I'll explain scratches.